So we now come to the main talk this evening, which is from Dr. Daniel Ives from Shift Bioscience, and we're going to learn how to live longer. Thank you very much.
Thank you. Thank you. Okay, so yeah, tonight I can't promise you'll go home and live much longer, but I can bring you onto the front line, just show you the progress that we've made so far, and it should be cause for hope at the very least. So the title slightly changed from what was put online. So it's leveraging cell simulation. So there's such a thing now called cell simulation, something that's a meaningful simulation, not just a meaningless abstract parallel. And using these new constructs, we can start to do huge numbers of experiments, specifically experiments trying to find how we can turn back the clock on cells and also turn back the clock on these diseases which accompany old age. So just to start with, just a reality that's all around us, that the fact that this is a worthwhile pursuit, not something that's futile. So rejuvenation, biology is a fundamental part of life. It's something that's all around us. So once upon a time, you were a baby. Before that, you were an individual cell, and that cell started from day zero. So even though that cell came from your mother and your dad and was around for 20 years, somehow you started from day zero. And this means there has to be a full reset available. If there wasn't, there'd be a generational debt of aging that you would take on from your parents, and you would live a shorter life and your children would live a even shorter life, and this doesn't happen. So either somewhere in the gamete formation or conception or post-conception, there seems to be some sort of rewind. And yeah, babies, and I've got a baby whale just as a second example. So this gives us hope that there is a safe rejuvenation out there that we can find. It's not something that's futile. And just to bring you right up to where we are at the moment, we're able to leverage, I'm going to use the term artificial intelligence only once, but we're able to use artificial intelligence to make cell simulations where we can then run experiments in those simulations and do vast numbers of experiments. So what I'm showing you here are the sort of distribution of 250,000 experiments where we overexpress 2 genes inside the virtual cell. We measure what happens as far as the age of the cell is concerned, and then we plot the distribution. So the 0 line is nothing happens, right, in response to those 2 genes being overexpressed. But everything to the right is these 2 genes age the cells, and everything to the left, which isn't very much, but is still substantial, is we've actually turned the clock back on these cells. So there's over 1,000 combinations of 2 genes that seem to reverse age. Which is really exciting. So we'll be testing the extremes in the new year from the simulation. So the very rejuvenative end and at the very aging end. And there's 2 things that might happen. One is that we actually show these combinations of genes do rejuvenate in the real world. But the other more likely scenario is that's the perfect data to make a better simulation. So we take that back to the simulation, say, look, you got it wrong, predict this is right, and predict this is right. And slowly you develop this positive feedback loop where your simulation is getting really good. So there is a path to getting there. But now I think what I want to do with I think it's 40 minutes I've been given, is talk to you about sort of the journey of the science and also the journey of the business. And I'm very grateful for the invitation. I've looked at some of the previous speakers, Demis Hassabis, how am I going to live up to that? Probably not. So I'm hoping this subject area, makes up for my own deficit as far as staff status is concerned. So science journey and business journey, I was going to try and separate the 2, but actually they're inseparable because one follows the other and vice versa. Science always being primary. So where did this all start, this crazy project? So it started with this eccentric Cambridge geneticist called Aubrey de Grey. I don't know if you ever bumped into this guy. But he is real. He's not from a movie. And in sort of mid-2000s, he gave a talk, TED talk, and in that talk he says, look, aging isn't inevitable. It's not fate, right? This is actually an engineering problem that we can solve. You know, if we just take a machine analogy, there's classic cars that are still out there well past their sell-by date. If we stay on top of the damage that accumulates in this machine and replace the parts, that may allow us to extend the lifespan of our own machine, our own human bodies. And more than that, he presented a framework for aging. So he documented a finite number of types of damage that accumulate as we age, and then proposed approaches to combat each of these types of damage. And this was compelling. So this sort of captured a generation of scientists, myself included. You know, I was working sort of biochemistry, molecular biology, and then Aubrey comes out of nowhere, like, we can end aging, and turns out you have a skill set that might be able to help with this. And it was just such a compelling draw. You couldn't say no. And better than that, there's another detail I forgot I was going to present, but something he presented in this talk that was so compelling was it's called longevity escape velocity. And this is the idea that if we can add a healthy year to your life every year that passes, you're not getting any closer to the finishing line. So we take a Wallace and Gromit analogy. You can keep putting railway tracks in front of you. As long as you put 1 year of track ahead of you each year, you're still on the rails, so to speak. And this is sort of compelling. 1 year of extra healthy life doesn't seem like such a big ask. It sort of reduces the perceived enormity of the task down to something manageable. So there's all these different arguments you can make on top of that. But yeah, all we need to do is add a healthy life year, because we don't want to be trapped in some dystopian, unhealthy body. We want to enjoy life to its maximum. So this was very compelling. And amazingly, there was a project sponsored by Aubrey here in Cambridge at the mitochondrial biology unit. So this was a whole institute in Cambridge that's focused on the energy producing organelle called mitochondria. This is one of the hallmarks of aging. The types of damage that accumulate with age is damage to your mitochondria. You just don't produce energy quite as well. And there was a project, and I actually applied to this project, but it had been discontinued due to technical difficulty, which doesn't mean there wasn't merit in the project. It was just very ambitious what was being trying to be done. But there was a side project in the same laboratory trying to achieve the same goal. And I didn't really have a plan B, so I decided to go with that. And yeah, I sort of entered my PhD the way that many biological science students enter their PhD. You get assigned to a protein. So after the decoding of the genome, you have all like 20,000 genes, right, in your genome. I think it's more than that, it's 40,000. I'm well out of practice. I'm just the finance and communications guy now. I can barely comprehend what my smarter scientists are saying to me. But when I entered my PhD, there's I think it's 40,000 genes. And you know, what are these genes? Like, we know they exist, but what do they do? How do they interact together? What's the amazing story? And so typically a PhD student's assigned a gene, and then you have to try and characterize that gene, like, what is it doing? And so, you know, I was assigned a particular gene, but the problem about this approach is very hit and miss. So you could get assigned a dud, right? And then that's 3 years of your life, and it's not coming back. And it's fine for your supervisor because he can just play the numbers, right? Just have 5 students, and as long as there's 1 hit, right, that's fine. But if you're one of the students on a dud, just doesn't feel great. So I was looking for a better way, and I basically, yeah, I didn't want this. My life, I don't want my life to be a gamble, which I think is reasonable. So I was looking for a better way. So something called computational biology was developing. Like, can we look across genes? Like, can we sort of zoom out from the trees and see the forest, right? Like, maybe we can look at a collection of genes which represent a pathway. Let's see what that pathway is doing. Yeah, just try and sort of start high level before zooming in, which was sort of the opposite of what was going on. So I started learning crude computational biology, and then there's something called gene set enrichment analysis, where you just take a group of genes and you measure if that group goes up and down. And surprisingly, even though I wasn't, you know, I'm not I wasn't remotely a computational biologist or competent to be so, I learned the tools, which were easy enough. And we were able, using this computational biology, to find a way to basically get rid of damaged mitochondria in a special model system. So it turns out that under certain conditions, you can make the undamaged mitochondria breed faster than the damaged mitochondria. So like the good guys win, they breed faster. And we still don't really know how this is working, which made it problematic to publish a paper because basically I'd created this method, didn't really reveal many secrets. It just allowed us to start doing things. So it was actually incredibly hard to publish. So even after we discovered a way, discovered a way to get rid of the bad mitochondria, it was 10 years before we could make a story out of it. So the journal doesn't want to know how you found some magic. So it didn't matter because I was sort of in that PhD to find technologies that could help solve aging. And when we'd happened across this, this was so exciting. Like we'd actually found something new. But the laboratory I was working for, they weren't set up to solve aging. That wasn't their goal. They were sort of working on rare mitochondrial diseases, which is very noble. But my sort of energy and passion was solve aging, like however unlikely that might be. So I basically decided to fund experiments out my own bank account, which is not advisable. I'm not sure why anyone didn't stop me doing that, but I did manage to pay for a sufficient number of experiments to start asking some more questions about, like, what is it we're doing and how can we make this better for aging. So this experiment here, I took commercially available fibroblasts from patients with idiopathic Parkinson's disease. So idiopathic just means we don't know why it's happened, which is the vast majority of these diseases that hit you in old age. So I took some fibroblasts from a patient with Parkinson's disease. Turns out they've got damage in their mitochondria. So this is just damage in the DNA that lives in the mitochondria. And when we apply a different type of drug that does have a sort of structural similarity to the previous drug, you can basically get rid of this type of damage in this Parkinson's cell. So number one, we didn't know where the problem was coming from in these Parkinson's patients. We find this type of damage. Number two, we can get rid of this damage. So it's like super exciting. And then I also, for a period, most of my day job was collecting saliva from people, which is a really messy business. I wouldn't recommend it, but in the saliva, there's actually white blood cells. You can collect the DNA, and then you can see is there damage in the mitochondria. So you can basically do experiments almost like just out there. And we were able to show in the saliva of people with Parkinson's disease there was damage in their mitochondria, but not in healthy controls. So healthy siblings and children share exactly the same DNA in the mitochondria because it's maternally transmitted. And anyway, we saw this, and that was exciting as well. So yeah, last point, which is really important. On the back of this data we were generating basically out my bank account and through donations, I think firstly from my family, and some just from kind individuals, and then I think where the buck stopped was I asked my girlfriend, "Will you give me your life savings?" And she said no, which was the correct answer. She's incredibly wise. She said you should go to an investor that can afford to lose it. That wasn't something that occurred to me immediately. So we were able to actually, like through sort of the network in Cambridge, connect with Jonathan Milner, who initially made a donation, right? And the condition of the donation was, if you actually manage to do anything with this small amount of money, I want first refusal on investment. So we went back to him with a step forward on the data front, and we were able to get our first check and establish the company, which we called Shift Bioscience. And then, I was working on this sort of narrow hypothesis. Damage to the mitochondrial genome is a big cause of aging. If we get rid of that, it might have a really great effect. It was actually Jonathan that brought my attention to the first impact of machine learning or artificial intelligence in this field. And this it's called the cellular aging clock, or DNA methylation clock, or epigenetic aging clock. So it goes by many names. But this is a technology that allows you to measure the age of any tissue in your body. So I could take any different type of tissue and tell you what the chronological age of that tissue is without knowing your birth certificate. I mean, if you're interested in the details, it's the application of relatively crude linear models. It's a type of machine learning to the methylome. So across your genome, individual sites can have a methane added or removed. And this happens with age. You have different sites that behave in different ways. So you can just run machine learning across all of that, and it picks out the sites that correlate with chronological age. So this was back in 2013, and Jonathan said, "Have you heard of this clock?" Because I was working in the field and I hadn't heard of the clock, to my embarrassment. But the moment we discovered this clock, we realized this was the perfect tool to test our hypothesis, right? We had this sort of damaged mitochondria. Is this really linked to aging, or are we just kidding ourselves? Is it like a hall of mirrors where this is all that we see, and therefore that's the solution to the problem at hand? And we ended up proving ourselves wrong. We actually ended up disproving our own hypothesis. So these mice are genetically altered to increase the damage to their mitochondria artificially. So they get about 2,500 times the damage that a mouse would normally have. And they actually get a human aging phenotype. They get this pattern baldness behind their ears. They actually have inverse eyebrows as well. So you get like balding above the eyes. So mice don't look like this when they get old. Humans look like this, well, not exactly like that, but they look more like this. So you're sort of inducing these human aging phenotypes. And then when we apply our drug, our mice are looking happier, right? They're not balding, and on the inside, they're actually much better as well. But when we measure the clock, the epigenetic aging clock, which is, if we just look at tissues plus control, which is basically these mice here, these sort of sorry-looking mice. Of course, there's lots of variation, but these mice are exactly the age they should be according to the clock. So they look much older than they should be, but the clock says they're exactly the age, right? So they, I think, what is it, 35 months. So they're not aging faster, right? As in terms of what the clock represents, which is real aging, these mice aren't aging any faster. They look older, which is really interesting. So it seems like the phenotype of aging can be accelerated relatively easily because you're using these different things. But whatever fundamental aging isn't accelerated, and we were working on a pathway and therefore trying to develop a drug to affect aging, and unfortunately this type of damage wasn't linked to aging, so our hypothesis was flawed. So we ended up disproving ourselves, and credit to Wolf Reich. Wolf Reich, who was at the Babraham Institute, which is just sort of south of Cambridge. He had the first mouse aging clock, and he actually was generous enough to embed one of our scientists in his lab so we could learn the method, take our own clock readings, and prove ourselves wrong, which in the scheme of things is actually something you want to do sooner rather than later, ideally before you set up the company, because otherwise it's more painful. But never mind, better late than never. And yeah, whilst we were doing that, others were also showing that not only can you slow down or stop aging, you can actually reverse cell age to zero, even from super centenarian cells. So there's a collection of 4 genes. They're called Yamanaka factors after their discoverer, Shinya Yamanaka, a Japanese scientist. These genes were originally used to convert adult cells into stem cells, but later on, with the discovery of these epigenetic clocks, these cell aging clocks, it was shown that the cells, whilst becoming stem cells, also rejuvenate entirely to day zero. So this is sort of incredibly exciting. It's like, I talk about natural rejuvenation. It's all very nebulous, right? It's like, okay, there's a baby and you can clone animals, they live a full lifespan. Where is it really happening? But here you've got 4 genes and you've got a really robust biomarker of aging, and it's going down to zero and it stays at zero. So like we've really got something by the tail here. But as is tends to be the case, the devil's in the detail. So the consequence of trying to reverse cell age is cancer. So it's therapeutically, you don't want to take an older person and make a young cancer patient. Like nobody's going to line up for that drug. And cancer is the very worst side effect. It's not like stomach ache or something. It's like the very worst thing, and you don't want to be putting that near anybody. So we've got a pathway, but it's nowhere near safe for use, right, for application at the moment. But this does hint at something really exciting. So, we disproved our own hypothesis as a company. Others were showing you could reverse age to zero. It was really promising. So I really needed to find a new direction for the company fast. And it's almost like no stone unturned. So I ended up relocating to Silicon Valley, not because it's some cliche that I wanted to follow, but the center of gravity is terms of investment in the field and interest in this particular field was in that part of the world. Aubrey had actually migrated over to that part of the world because that's where you could get more support for the science at the time. Jonathan was about the only high net worth individual in Cambridge that was as engaged, if not more engaged, than I was about this science. He actually came to this, he gave a CSAR talk, right? He came here. I think he derailed his entire talk to talk about aging, which, you know, I sort of give him a lot of credit for. And there's a nice circularity later I'm going to get to. So I relocated to America. I got to drive a pickup truck, which was very nice. I'd always wanted to do that, be a real American. Yosemite is beautiful. But most of this, I think 3 to 4 month sabbatical was just pain and learning. I thought I could go over to America, just ask for 10 times more money without leveling up. And it turns out there's way more competition over there. There's things that you just don't know are happening until you go over there. But it is some weird sort of qualification. If you go out there, it's almost a statement of intent. People do treat you differently rather than like a Zoom call. You're just tuning in. It's like no cost to you. Going out there does mean something. And the best thing that came out of this trip, not the investor criticisms. I mean, the learning was good, but it was this. It was an event run by X Prize. So I don't, if you're not familiar, X Prize is like this prize giving organization. You create this huge prize pots, and it encourages private enterprise to create solutions to things that otherwise wouldn't have an economy. So imagine you have a 100 million prize pot, and you could go to an investor and say, oh, look, you could just give me 2 million. And I'm going to win this prize, and you can get a big return. So there was I think they did a prize for private spaceflight, and it had some really sort of visible, exciting results. But X Prize were interested in creating a prize for longevity, right, or increasing lifespan. And they held this workshop as like a due diligence exercise, like, you know, what's the finishing line going to be? We can't have it too far out because then nobody's going to get to it, and it's all disappointing and a big failure, and everybody's sad. So it needs to be something that's within reach. And so I was invited to this. Fortunately, Aubrey was there. Many of the great scientists in the field were there. And the first discussion was, we can't hold a competition if we can't measure aging. And I'd just been using these aging biomarkers. So I put my hand up and I said, look, we can measure aging. It's called the epigenetic aging clock. It can predict any age from any sample across your body without knowing your birth certificate. And it turns out Steve Horvath, the inventor of the clock, was in the room. I just didn't know he was there. So he stood up and he said, oh, I'm actually here. And I'm like, that's fantastic, Steve. You can take over. The conversation became much more educated very quickly. And basically, I managed to, strike up a conversation with Steve, talk about what we were doing. He was impressed that we had that much faith in the clock, because these products of machine learning are like black boxes. You have to have a degree of faith. It's like an alien technology. But if it behaves in reliable and predictable ways, and according to gold standards, you can lean on it. So Steve was impressed by what we'd done, and he said, actually, if you're trying to find a new hypothesis, right, if you want to target aging to reverse it, and we want to rejuvenate, and you want to find that biology, you need to talk to my favorite collaborator called Ken Raj, who was back in the UK at Public Health England, which I think recently has become the UK Health Security Agency after COVID. And Ken was the only scientist in the entire world, as far as I can tell, that was asking the really interesting question, which is, what is this clock? What is this epigenetic aging clock? What's the biology driving it? Why is it so, accurate? Like they're the really interesting questions that might suggest drug targets. So I ended up going back to the UK, meeting Ken. He's my favorite scientist to date. Just matter of fact, like, this is what we need to do, and quite quickly we're on the front lines with Ken, and we found a new direction. And that direction was, we wanted to do a genetic screen for aging. So we wanted to knock out every gene in the genome systematically, and then measure what age was doing according to a clock. However, using the current aging clocks, this would take, I think it's 50 years, right? And it would take multiple billions. It just wasn't viable. So if we were going to do this genetic screen for aging, we needed a high throughput clock, also known as a single cell aging clock, sort of in our field. And this is the connection with CSAR. So there was a talk by Jonathan Milner. In the audience was a PhD student called Brendan Swain, who was attending. He actually wanted to hear about the businesses Jonathan had set up. But instead, Jonathan started to talk about aging and not his businesses, and Brendan was really annoyed about that. But he did learn about what we were doing, and he actually reached out and said, look, I need to do an internship. You don't need to pay me. Can I do it with shift? It sounds interesting. And Brendan was one of these rare individuals, not so rare in Cambridge. You you give them a task, and not only do they complete the task, they do something beyond the task that you didn't even ask them to do. And so every time Brendan did this, we gave him a more difficult task as a reward, and he just kept going and kept going. And after a few rounds of this, I was struggling to keep up. Eventually, his internship ended, but we stayed in communication. And then later on, after Brendan's PhD came to an end, he decided that there was more exciting science to be done within sort of what we were doing together. And so, just in brief, Brendan was able to apply the same types of machine learning that were used to create the original clocks, to create a single cell aging clock. So being able to say what the ages of individual cells using gene expression, not methylation data. And this actually allowed us to do a genetic screen for aging. And this is just a figure showing you, this is publicly available data from mice. So each dot is an individual cell, and we can basically predict the age with a really, high correlation, so 0.92, where 1 is perfect. And you can see there's a distribution of age in the samples. So not every cell in a sample is the same age. Some are older, some are younger. So anyway, this was a big breakthrough, and I think he was the first person to be able to do this. Now, what was really interesting was that because this aging clock was built out of a list of genes, you could look at these genes and see like what is going on, right? Like, what is this clock? The original clocks, you couldn't see what the cogs were, but with this clock, you could see what the cogs were. And you could look at those cogs, and lo and behold, some of those cogs were biology that, sorry, this point is not very accurate, but this biology was known aging biology. There was like these positive controls suggesting whatever this algorithm was, it was meaningful. So it's like by running this machine learning, we had this black box decoder, like here's this mysterious process, and now we can find the genes responsible for it. So we called it like the driver clock. So, we create a clock that correlates, but it also reveals its secrets, like the underlying drivers. And even better than that, you can apply this to other black box processes in biology. So rejuvenation by these 4 genes called Yamanaka factors. We applied this method, and it also it picks out biology that you would expect, things that interact with these Yamanaka factors, but also novel biology. And so on the back of that, in 2022, we geared up to test the predictions, like have we found this rejuvenation pathway using this algorithm? So we basically geared up to put these different genes into cells, see if they rejuvenate. Okay, and so during this time we received quite a lot of investment, because I think principally because a large company that was absolutely focused on what we were focused on announced. So lots of people with credibility from outside our field assigned themselves to this problem, and therefore imported credibility to the field. And so all of a sudden, what wasn't previously a business opportunity became something everybody wanted to get hold of. And we were like instantly oversubscribed, and we closed investment in like 2 weeks. It was crazy, like to be on one side where you can barely raise anything, and then be on the other side and you just raise all this money all of a sudden. So no technical breakthrough, right? Simply everyone decided this was real, right? That was the difference. And then what happens is once you raise investment, it's all very exciting, but that money has to flow through you, right? Like you have to action it responsibly, and that's primarily a recruitment and delegation issue. So you have to find lots of people relatively quickly that are going to be around for a long time, and they've got to mesh together well as a team. These aren't things that you're taught as a PhD scientist, right? You sort of have to get to grips with it very fast. So something that we learned during this process is that you can recruit a team very quickly, and there's a sort of honeymoon period where everybody's very excited, but quite quickly the high performers get annoyed by the low performers, right? And so your inclination is to try and help the low performers, but actually that's completely upside down. You need to do everything you possibly can to keep those high performers, including getting rid of low performers, because otherwise it threatens the whole sort of existence of your company. So like having a turnover is actually a feature of a team that's intent on making something happen, not just sort of laying into the problem, getting comfortable with it. So that's hence the squiggly, sort of having to go back. It's just hard to put all the people together and make it work. And now we're a larger team, so team of 9. It's not a very big team, but historically we were a very small team. It was just me to begin with. And we raised all this investment, and we were going to test this, this prediction, right? Can we rejuvenate cells? And we couldn't. So our method didn't it did find new genes. We found new genes, but the wrong genes. So we managed to find more cancer-causing genes, which, sort of, if you continued with that, it's like the light bulb, right? Find a million things that don't work. It's probably, and then you find the one that works. But bioscience is very expensive. You just can't go for very long with that type of approach. So what we can do is, so this is basically, we deliver the amino factors to cells, and each dot is a cell. And if it's red, it's basically in this sort of cancer-inducing state. And we deliver our novel genes, and then we find some of those cells become cancer, and it only takes one cell. So it's interesting, we can find things that are new, but we want to find things that rejuvenate that are new, not new ways to make cancer, which should be obvious. And so in parallel, we were investing in a technology that would allow us to systematically find those genes, not just sort of create a single prediction and hope it works or not work. And this was a far more accurate single cell aging clock. So previously, Brendan had been able to create these clocks from public data, but to create a really accurate clock, you needed to invest in a big data set. So this is the clock we were able to create. It's you can use it in human fibroblasts in the dish. So this is the high throughput clock versus the gold standard epigenetic clock. So yeah, basically very high correlation. We had to create, I think it's the largest data set of human fibroblasts that we're aware of. Maybe this is out of date. So there's over 100 cell lines. So each one of these orange dots or blue dots is a female or male sample. And then with these types of machine learning products, you need to train the clock, and then you need to test the clock in a separate data set. So this took about 1 year and 1 million, which is a long time and a lot of money. And this is, I think this was the visual for the talk today. So this is actually every single one of the cells from that data set clustered according to their similarity as far as gene expression is concerned. So it happens to make a really nice picture. I don't know what it means. It's a bit like looking at clouds. Maybe it means something to you that's different to what I see in it. But each dot is a single cell. You can see sort of the vast number of cells, and that's where all the money's going. But it is the largest data set. And the really exciting thing about this clock is what happens when you combine it with what we call cell simulations. So you may have heard of ChatGPT. If you haven't, I'm not sure where you've been, but I had this dawn chorus like ChatGPT, AGI, why are you going to have a job? And I was like, oh, I've got the data set. I can eke out another 2 weeks by tolling access to the data set. The AGI won't kill me immediately. But you had all these people really worried, like too worried about what was going on. So the same models that can learn the relationships between words and create these sort of amazing context-aware chatbots, you can also use those to learn the relationships between genes inside individual cells. Basically create gene networks that are basically simulated versions of those networks. You don't just need, you can do this without transformer models. You can use other things like neural nets or graph neural nets. But the main thing is that there's like an abstract simulated version of the cell available, and this clock is built in such a way we can use it in that simulation. And this is a really big deal, because if we want to find new combinations of genes that rejuvenate, if we're just looking at combinations of 3 genes, there's 365 million combinations of 3 genes to test in the universe that we're looking at. And if we tried to do that in the real world, it'd be 50 years, it'd be multiple billions of dollars. If we can use simulations, we can reduce that down to a couple of years, and we're in sort of tens of millions of dollars. So it's really enabling. And so just recently we ran our first simulations. So this is overexpressing. Yeah, I showed you this a bit earlier. So overexpressing 2 genes, and we're really excited about this subset of genes here on the left-hand side. So we're going to be testing that, and also on the right-hand side. So yeah, stay tuned. And then what's ahead of us? There's this huge number of experiments we need to do, like 365 million experiments represented by this square. It's going to take us about 2 years, and we want to basically create, we call it the landscape of rejuvenation. So imagine in this landscape, each peak is a combination of genes that safely rejuvenates. We want to find Mount Everest, K2, Table Mountain, all of the different peaks, because then we can basically find the best combination as far as a drug's concerned, right? Like, something we want to use in the body and eventually across the body. And yeah, to be able to complete this exercise, we've had to move labs. So recently, I think until recently, we were on the Addenbrooke site. So there's lots of biological research institutes up there. We've now moved to the Gurdon Institute. They've made a little space for us. We're going to grow the team from about 9 to 22. So each of the blue roles are people that are already with us, and then each of the grey roles are new roles we're going to add. And we've got this nice laboratory here. It's a really nice place in Cambridge. Yeah, for what that's worth. And then, yeah, something that's also happened is learning how to communicate properly, because to try and make these things happen, you basically need to be a translator to lots of different types of people that want to help you but don't know how to help you. And that might be people from finance or management or health and safety. You need to be able to connect with all these people. And when you finish your PhD, at least for me, you're sort of this scary creature that talks in acronyms, and you can communicate directly to other PhD students, but not really anybody else. And you might be fortunate, you might meet a scientist who's become something else, and so they put up with your poor communication. Outside of those narrow scenarios, you really need to sort of listen, be aware of somebody, see where they're engaged. I remember so many times people's eyes drifting. So just make sure they're still there. I remember there was this lady, and I was very energetic and passionate about what I was saying, and then after 20 minutes, she interrupted and just said, "I don't really understand English. Can you start?" So I just failed anyway. It's just simple things. So yeah, like some of these visuals, trying to connect with people like in a meaningful way. There's also this movie. This director's reached out. He's creating a movie called Last Generation to Die. So the plot is it's the first clinical trial for a drug that can reach across the body and basically turn the clock back. And it's all about the human story. So like all of a sudden everybody wants access, and the person running the trial, it's actually their dad that she gets enrolled against regulation, and she overdoses him because it's getting really, anyway, it's a really amazing human story. And this guy reached out, he wanted to bring the science right up to date. I was quite happy to contribute, because it turns out film is an amazing tool to take somebody out of all of that distraction and systematically put them through sort of a thought process and take them to quite an informed place. So when you sort of view it in that way, it's a very sort of high leverage contribution. So this still hasn't been made because it's an independent film and sort of financing is hard for everybody. But it's an amazing script. I hope he publishes it as a book. Sort of really sort of paints an amazing sort of turn of events. So yeah, learning to communicate effectively, that's sort of part of the business journey. And then also just on the business side, you can't develop a drug to slow down aging or reverse aging. Like, there aren't people going to the doctor right now saying, "I'm old, can you help me?" That's not classified as a disease. And I think there's something called a good innings, right? So a good innings is you've had a full and healthy life. You should be grateful, yeah. Which I think that is the general understanding, and I think that is correct. So if you try and reclassify being old as a disease, that does sort of an injustice to anybody that's got anything so bad, worse than a good innings. So I think what's starting to happen is, if you want to go beyond a good innings, that's called augmentation, right? And then anything on the other side of a good innings is, obviously not great, right? It's getting a certain disease. But it turns out the same biology that can affect the age of the cells can actually affect the diseases. So you can sort of reverse multiple disease states across the body using these same sets of genes, which is sort of incredibly lucky, because it gives us an economy that allows us to keep pushing these types of drugs out there. So when we try to run a first principles analysis of what disease we need to target, the I, so age-driven blindness, is optimal according to a bunch of metrics. The main metric being that others have used the unsafe rejuvenation genes to reverse different measures of vision loss. I think there's a mouse glaucoma model and a non-human primate laser-induced blindness that you can sort of reverse with this type of approach. But remember, these are cancer-causing genes, so we just want to follow up with something a bit safer. And then just sources of motivation, because a lot of this is very hard. You don't get a good result very often. It's mostly painful. It's just, oh, how much money have we wasted? How can we not do that in the future? So motivation number one, which is like the stick. We don't have very long. So each one of those squares is a week of your life. So 52 across, 90 down. I fill out mine at the beginning of every week. It's sort of helpful. It's helpful anxiety for me because I'm working on the problem. I wouldn't recommend this for everybody, but we haven't got that long. And so if this isn't within reach and we're complacent and we don't sort of move with 100%, we're not actioning this, that would be tragic, right? To just be so close, but basically miss out on something really amazing. So that's the stick. But this is the more exciting bit for me personally, which is if you can control biological age across your body in a meaningful way, your sort of future becomes very different. So just bear with me here. I think Brendan rolls his eyes every time he hears me say a great delta of time and space. So hopefully this goes down well. So this is the history of the universe. I think there's people going to tear me to pieces later, but Big Bang, expansion, initial expansion, a lot of time, slow expansion, and there's a lot of time to the right, way more than the space dimension. So our existence at the moment is like a single pixel in space and time. So just born out of nowhere. So all this stuff's around us, like what is even going on? Like there's how many layers of reality we don't have perception of. Then we're gone after 100 years, like who knows what's next? It's pretty bizarre. Now, the moment you've got biological age control, things change pretty dramatically. So now you can control the biological age of your cells. I'm not forcing you to do this. You don't have to stay in life prison if you want to go to the next bit. But you now have, well, I would say, a less risky option, which is to stick with what you've got. And because you can survive time, you can also survive the journey across space, which takes an incredible amount of time. So basically, the future starts opening out into the time and space dimension to the extent that the entirety of the future eventually becomes synonymous with your future, which is just a completely different forward-facing view. Like at the moment, we don't even talk about death because it's that bad. It's like nuclear weapons versus normal weapons. It's just so bad, you just got to put it out of your mind. Like death is that bad. I've been to a couple of funerals recently. Everybody knows how bad it is. And you're just trying to keep yourself together, right? During these, you sort of turn to different things. It's that bad. And to not have to deal with that massive downer and sort of suppress that into your darkest parts of your conscience, like just wake up on a Monday and it's great. It's like, oh, I'm not going to die. Fantastic. That moment you realize as a kid, it's like, it's that feeling and then you just suppress it. So imagine having that weight lifted. This is going to take ages to get there, right? But I'm just saying, like, this is a very different future when you do get there. So for me, this is personally exciting. Like, we have a board at Shift, which is like, what would you do with the extra time? So I'd like to fly my FPV drone, which is a bit strange, but it's like an aircraft and you've got a camera on it and you strap a screen to your face. I enjoy that. I'd like to do some tornado chasing, adrenaline hit. Like everybody's got their thing, but if there's things that you really like in life, right, and there's more life available, this is a really nice thing. So yeah, maybe if there's a bunch of space infrastructure, we can actually get to something that's like supposedly better than Earth. So maybe it's like the Caribbean across an entire sphere. Wouldn't that be nice? But you can actually, there's a completely different menu of options, right? It's not just the set menu we have at the moment. And I don't want to seem ungrateful for a good innings, but I do find this very exciting. And then just some credits, because I just get to talk about all the interesting stuff. All of the really difficult and finicky and tedious stuff is done behind the scenes. So first credit to the team. Andreas, Raya, Charlotte, Kate, Samira, Brendan and Lucas, and Steve, who's actually my dad. He got taken along for a ride, took him out of retirement. Our investors, Jonathan, F Prime Capital, Kindred, and then our advisors, Tony from the Gurdon Institute and Leo. And this is a product of machine learning. So I think if you use ChatGPT on visual mode and you say, I want a aging clock, cellular aging clock with DNA featuring actually spits this out, which is sort of incredible. So I think that's it for the time being. Looking forward to any questions.
That was fun. It took us in places I didn't think it was going to go. Your previous slide with this sort of expanding universe, was that an investor pitch slide?
No, I'm not allowed to give this investor pitch. So there's almost there's the front end version of this, which is you target diseases, and then there's the back end version, or the, I don't know, it's like the unofficial version, but it's, you know, it's sort of things like this, you know, the application to sort of space. There's also, I don't know if you've heard of Brian Johnson. He's a high net worth individual in the States, has a personal budget of $2 to $3 million per year to keep himself young. So there's these individuals that are taking personal medical sovereignty, flying themselves out to, I think it's a Caribbean island where they can do anything to you, and they're taking the risk, and they want a gene therapy across their whole body, right, despite the fact there's been no clinical trial. So like there's all these sort of orthogonal routes, but that doesn't create an economy which can actually make this real. So we have to go down the normal routes, otherwise you're marginalized. And this isn't going to happen in a spectacular way. If you get this done the official way, it can go everywhere else.
Great. Questions? Everyone is stunned. So what happens to aging of sort of neurodegeneracies and so on, if you manage to solve the business of staying alive? Do we avoid Alzheimer's and other conditions?
Yeah, we don't want to. What's happening with increased lifespans at the moment is the unhealthy part of your life is increasing. The healthy part is staying relatively constant, but the unhealthy part is increasing, and that's not good for anybody, right? So if we're going to be doing this, we're fortunate that the same biology that helps us solve this problem can actually affect diseases. It seems like we can rescue cells from any bad state, whether that's disease or old age, back to a healthy, resilient state.
So everything gets pulled back together, so it's a sort of win-win.
We think that's on the table.
Right. Okay. Yeah. We have a question. Yes.
Fascinating, really. And rather than looking at rejuvenation, we're told by marine biologists that if you can slow down cell growth, I mean, I understand there are some sea creatures now well into maybe 200 years old, something like that. And I just wondered how that might relate to the work that you're doing in terms of slowing down cell growth, or being able to bring it up to a certain level and then slow it down so that aging is retarded in that way.
Yeah, we call this the low-hanging fruit, because nature's discovered ways to slow things down, and there's sort of there's pathways that kick in during starvation that help to slow things down as far as aging is concerned. The problem about a lot of these low-hanging fruit is that you substitute some sort of enjoyment or utility for something that is painful and uncomfortable. And so it's the great irony, right? You could live a long time, but it's mostly because it feels like a long time, right? It's even though you're living longer, you don't want to be, you want to check out early, even though you can.
But this sounds like academic tenure to me.
So what we want to do, we want to maximize the utility of life, right? And at the same time increase life and have maximum utility, which is enjoyment. Hence, what are the things you enjoy doing? Can you continue to do those? We don't want to trade. Don't want to be a zero-sum affair.
I can quite appreciate the possible advantages of slowing down aging, but I'm a little bit more concerned about trying to reverse the situation with de-differentiation and the potential for malignancy arising. Can you elaborate a little on that? Thank you.
Yeah, so this is the great challenge. This is the commercial and scientific pursuit, which is like, can we separate the pluripotency problem from this rejuvenation pathway, from this disease reversal pathway? All we know at the moment is there's 4 genes, and they activate all of these 3 things simultaneously. There is evidence we can start a separation between the rejuvenation and the pluripotency. We don't know we can reach 100%, but I think for this to be therapeutically viable, it needs to be 100% because it only takes one cell. Like imagine you're trying to do this across the body, how many trillions of cells there are. So you need to be zero. Ideally, you're an anti-cancer agent whilst you're doing this. Like if you aim for that, then we're more likely to succeed. So that's the principal problem. If we can't solve that problem, then this is just going to be confined to the dish.
Please, Robin. Yeah.
Thank you. We hear in the sort of popular press that different organs in our bodies have different ages. Is that a credible idea? Does that have any bearing on what you're trying to do, or insights and how you might do it?
Yeah, so the brain is surprisingly, the brain ages the slowest. I don't know whether it has some privileged status, probably does, but it ages the slowest. Breast tissue ages the fastest. So I don't know exactly what that means, you know, why that's the case or why that matters. And then you do have sort of differential aging across the body. There is an argument, which is if you're gonna rejuvenate one system, you sort of need to rejuvenate other systems. So imagine you have a vehicle and you install a massive engine in that vehicle that wasn't designed for, something else is going to break, right? So like imagine you rejuvenated your muscles, but you didn't rejuvenate your heart. It's a problem waiting to happen. So I think there will need to be some sort of sensible coordination, but it doesn't seem like, it seems like this is manageable. Yeah.
Great talk. What about aspects of aging that don't have a genetic undertone? So like, for example, where Kevin Chalou showed that the extracellular matrix stiffness can influence sort of aging in cells and things like that. How can you address that aspect of aging?
Yeah, so the technology that I'm talking about, it operates within a narrow window. So that is, there is a cell that's still there that we can now revive, despite its circumstances. Yeah, despite its circumstances. So extracellular matrix is something that surrounds the cell. So basically, there can be a bad matrix that actually makes a cell behave old, even though it's a young cell, right? Sort of the matrix is doing something. That will be a challenge, and there's evidence you can basically over time you can sort of recreate a younger matrix, right, and not sort of fight against the tide. But there is a real discussion. I think I almost point to another problem, which is we can't revive dead cells, right? If you've lost cells, which you do, like as you age, you lose cells in your brain, across the body, and we can't revive dead cells. So the cell still needs to be there for us to bring it back. If the cell's been lost, we need to replace that with a new cell. So like replacement's going to feature if we want to create a comprehensive solution. But there's a bunch of, this isn't a perfect solution, and we fully acknowledge that, and there will need to be sort of secondary solutions to fill the gaps. Yeah.
Got a question at the back.
Thank you very much for a great tour d'horizon, a call to arms. I was just reminded, you talked about Silicon Valley, and Steve Jobs in his day, one of the great wunderkinds of Silicon Valley, did of course say that death is probably one of life's greatest inventions because it clears away the old and makes room for the new. And here are you, an embodiment of new thinking and originality. Isn't it going to be terrible if we leave an awful lot of old brains and old minds getting in the way of the new and the vigorous?
Yeah, so I'm gonna sort of recycle an argument from Andrew Steele. So he's a science communicator in the UK. He published a book called Ageless. So in this book, if you imagine a society where aging doesn't exist, right? So it's just imagine a parallel universe where aging doesn't exist. You're born, you're perfectly healthy. Now in that society, would you invent aging to get rid of institutional stasis, right? It seems like a bit of an overkill, right? You want to take out this guy, but the side effect is it kills everybody else. It's actually quite like nuclear weapons. It's like, I want to get rid of this problem, but the side effect is I'm going to get rid of everybody else. So all these solutions that are like, all these things where aging is elevated as a solution, right? If you were to invent it in an ageless society, it just seems barbaric, right? There must be a better way, right? Like we can create a new structure, and we can, you can write a a code, which is you only have 5 years. We don't have to kill everybody in the process.
I'm getting.
I have a last question.
Yeah, fantastic journey. You've talked a lot about genes. Do you see, and there's two parts of the question. Do you see the product being a gene therapy, or are you going to use those, which is going to be very difficult to develop, or are you going to use those to discover small molecules, which is another difficult to develop? And related to that, when are we going to get it?
Yeah, so timelines are problematic, but let me just attempt it, right? So we think it's 2 years until we have this landscape. So we sort of know the genes that we're going to be targeting. And then to your question, what's this going to look like? So, you know, there's something called mRNA drugs that are available now. So at least in the eye, we can use these sort of gene therapies, right? So there's a narrow path to using our knowledge immediately in the eye. So I'd say 3 years for preclinical. I'm not the expert in this side of things, but 3 years for preclinical, 2 years for each stage of trial. So it's 6 plus 3 plus 2, which is 11. So I've still got a bit of maths left. So that's sort of a very narrow path. And then there's a question of like, okay, we find these genes, but maybe there's biology that's proximal, that's inherently druggable. Yeah. So we can actually find molecules, and molecules are much better. We can get to far more of the body with molecules just because they're smaller. So, you know, when we get there, we might realize there's a better path, but there's also something we can do which is very opportunistic, which is right now, we can take clinical stage and clinically approved molecules and just use our high throughput clock and just screen them. Because wouldn't it be nice if there was a molecule just sitting there, we never had the capacity to look, but now we do. And we sort of have to do this out of responsibility because, you know, like I would, you know, be asked to leave my job if I hadn't have done that, right? It's such a, it's like everything else is procrastination if you haven't just checked the low-hanging fruit. We don't expect there to be a molecule that already exists, but we should definitely look, and you know, there might be, right? And then things are going to move a bit faster, but I think we should be conservative and assume an uphill battle, you know, but sort of 11 years would be, it's not just my guess, it's about 11 years until we get this to one part of the body. But once that works, it becomes really exciting because people are thinking like, where else can this go?
So I'm worrying about your little map of the weeks of your life left and whether you sort of hit the bottom of the screen by then. But yes,
yeah,
there we go. Yes, yeah,
yeah. So I'm about here. I'm about 37. So yeah, I've got. I mean, I don't want this to take 30 years, but if it had to, I mean, if you guys spread the word, right, we can make this happen a bit faster. I mean, it's in everybody's interest.
Okay, I think that's a wonderful message to take away. Yes, that we've got to spread the word. That was completely mind blowing. To be able to pull cosmology into life extension is remarkable. Thank you so much.
You're welcome.